Good evening, everybody, and welcome to tonight's webinar. We are so pleased to have Dr. Michael Borrie join us tonight. It has been way too long since we've had you speak at McCormick, Dr. Borrie, so thank you so much for coming. So Dr. Borrie is a geriatrician and professor at Western University, medical director for the Aging Brain and Memory Clinic, an investigator with the Cognitive Clinical Trials Group, and program director of the Southwestern Ontario Regional Geriatric Program. He's a busy man. His research interests include predictors of conversion from mild cognitive impairment to Alzheimer's disease, including sensitive neuroimaging techniques and randomized controlled trials of new treatments for MCI and Alzheimer's. He is also the president of the Consortium for Canadian Centres for Clinical Cognitive Research. I'd like to welcome Dr. Borrie.
Thank you very much. Good evening. To join this webinar on what's new in Alzheimer's disease research. First, I need to go through my disclosures. On the left are the funding organizations, CIHR, NIH, which are national organizations in Canada and the US funding our research, and through the Alzheimer's Society Canada, Physician Services Incorporated, and Lawson Health Research over the years have contributed funds. On the right side, we have clinical trials funding for clinical trials we've conducted, as well as companies listed on the left who have provided clinical trials funding, and I have also been a consultant and has done programming for education of family physicians. All of the funds that come to our clinical trials groups are received by Lawson Health Research Institute, and they fund and support the research group, the coordinators, the research assistants to actually be able to conduct the research. It's not anything that is coming, and I'm not funded in any way by this. The objectives that we want to cover today, this evening, is to review the lifetime risks for dementia and how to modify them, and the evidence, some of the evidence for that, review the Alzheimer's disease drug development pipeline up to 2022, talk a little bit about the theory for Alzheimer's disease and the development of biomarkers, and these are biomarkers measured both in the cerebrospinal fluid, blood, and also imaging using positron emission tomography or PET scans to detect Alzheimer's disease. Talk about the latest results from the lecanemab randomized controlled trial published in November of last year. Talk a bit about the potential side effects from removing amyloid protein from the brain, and I'll explain what that means, and then review some of the current intervention as well as observational studies being conducted at our site and in Canada. This describes the spectrum of cognitive decline, and you can see at the left there is a group of people who we characterize as being supernormal, people over the age of 80 who score the same as 20-year-olds on standard memory tests. Normal age consistent loss describes most older individuals, people who have no concern about their memory, and their scores on memory tests are in the normal range. So if we use a standard memory test in a clinic, such as the Montreal Cognitive Assessment or MoCA, they would have a score of 26 or better on that scale. And then people with subjective cognitive decline are noticing changes in their memory, and secondly, are concerned about it. And they have scores on a test like the MoCA, 26 or better. People with mild cognitive impairment notice the changes, are certainly concerned about it, but their scores on the memory test are below the cutoff, and in the instance of the MoCA would be less than 26 out of 30. But their instrumental activities of daily living are entirely intact. So the ones that are listed there are examples of cooking, finances, and managing medications. Dementia, on the other hand, is determined by information that comes from the family of people who notice changes in their memory, they have impairment on memory scores, and family will report that they have problems with one or more of the instrumental activities of daily living. And that's how we get to a diagnosis of dementia. It's information that caregivers and family members provide. And that's really important to remember. At this point, dementia, that's how we define dementia. Across the bottom, you can see that in Canada today, people 65 and over, 8% have dementia, 17% mild cognitive impairment, combined 25%, with 75% more or less falling in a normal range. And then at 85 and over in Canada today, 1/3 have dementia, 1/3 mild cognitive impairment, and 1/3 fall within a normal range. So it's dementia and cognitive impairment is common in older age, but it's not caused in itself by old age, because there's 33% of people who are older who don't have dementia. Once a person gets to dementia, you then have to ask, well, what is the underlying disease that is causing this dementia? And the most common is Alzheimer's disease. Vascular dementia is most common due to small strokes. And sometimes people may have a combination of Alzheimer's and vascular disease. And that can be determined using imaging such as an MRI scan. And then Lewy body disease can also occur. Often it arises in combination with Alzheimer's disease. And then there's frontotemporal dementia being less common. And then there are other dementias listed on the right, which I won't be describing tonight, but nevertheless acknowledge that there are many causes of dementia. This really describes the potentially modifiable risk factors for dementia during the life course. And you can see each of the risk factors highlighted in the bubbles. And you can see within the bubbles a percentage. And this percentage represents the proportion of people with dementia, the proportion that you could reduce the prevalence of dementia by eliminating that risk factor. So for instance, if you could totally eliminate hearing loss, you might reduce the overall prevalence of dementia by about 8%. So you can see this is accounts for about 40% of the risk factors for dementia are modifiable, but there's still another 60% which is not accounted for using this data, which has developed this database. This was published most recently in 2020 by Livingston and colleagues in The Lancet. So there are various strategies to be able to reduce the risk and manage cognitive impairment, and I've just listed them there. You can read them, and these are ones that we commonly discuss with people when we see them in the Age and Brain and Memory Clinic, or when family physicians might talk to people about reducing the risk of dementia. And you can see some examples of some of the things that people might do under each of those items. So regular exercise is probably the most important one, paying attention to diet, getting adequate and particularly restorative sleep, having some forms of mental stimulation, engagement with people. Certainly we're aware that during the pandemic, socialization was diminished in both people with cognitive impairment as well as older people without cognitive impairment, as well as younger people, did have a significant effect on their cognition. And hopefully this is improving now that people are able to socialize more. Education is important. Certainly lack of education at a younger age is an important risk factor. Fortunately, in Canada, where we have a strong education system, that's less of a concern for younger people. Managing stress, avoiding brain injury, dental hygiene, flossing and brushing to avoid the effects of poor oral health affecting various diseases, diabetes, aspiration pneumonia, which will have a compromise on people's cognition, particularly if they should get delirium as a consequence of that. Air pollution, and we're in an environment now where I think we're going to see some further evidence of this, because right now, today, Canada's on fire, and United States is suffering along with us. Even Norway today is suffering. So this is a serious issue and something that we're going to have to pay attention to even more. So more strategies, alcohol and drug reduction, the Canada guidelines this year, 2 standard drinks per week is the upper limit of a safe drinking amount for many diseases, cancers, as well as cognitive impairment. But certainly people that we see who already have some cognitive impairment, we encourage cessation. There are a number of vascular risk factors there that cause stroke. These need to be detected, treated, and managed. They're listed there. Kidney impairment, depression, both treating using non-drug as well as drug treatments, and then compensating for hearing impairment. This graph reflects a study that was conducted over about 12 years. You can see that across the lower part of the graph, the x-axis. And on the vertical axis, you can see the probability of remaining Alzheimer's disease or dementia-free over that course of that 12-year study. And this was published in 2009. And with this observational study of cohorts of people, they followed them during this 12 years and got them to characterize their activity by high, medium, or low, and a high adherence to a Mediterranean-style diet versus a low adherence. And you can see there the 2 groups that stand out are the ones with high physical activity and high diet score, and they have a probability of remaining Alzheimer's disease free over the 12 years as about 80%, compared to the group of low physical activity and low diet score, and they're less than 60% probability of remaining Alzheimer's disease. So this is an observational study, very difficult study. You couldn't do this as a randomized controlled trial. You can't randomly have people to have a poor diet versus a good diet, because everyone's going to want the poor diet. So we have to use these observational studies. However, there are purposely designed studies where there are multi-domain interventions. And this was an important study published in 2015 in The Lancet by Mia Kivipilto and colleagues from Finland and Sweden. And they did this Finnish geriatric intervention study to reduce the risk of cognitive impairment and disability, the so-called FINGER study. And out of this has been a worldwide initiative called the Worldwide FINGER. So many countries are now adopting these multi-domain intervention studies. This particular trial was a 2-year randomized controlled trial with participants recruited age 60 to 77 who were at risk of cognitive impairment, with 4 different interventions. Nutrition was one intervention, physical exercise, cognitive training, and vascular monitoring in the intervention group, and they got individualized counseling and instruction on those things versus health advice for the control group. And then after the 2-year period of time, they found there was improved cognition and reduced risk of cognitive decline for the intervention group. So this was a very important outcome. Switching now to talk a bit about Alzheimer's disease. This is a representation of what Alzheimer originally saw over 100 years ago, and he described plaques and tangles. And you can see on the left a normal appearing cytoplasm, the brown tissue in the brain, and 3 normal appearing nerve cells. Now we have billions of nerve cells in our brain, and these nerve cells are talking to each other. And here we on the right side, you have in the cytoplasm the brown plaques of amyloid plaques being indicated there with the pointer. And you can see that there are the purple neurons, don't look as healthy as the ones on the left. And in this representation, they have what we call neurofibrillary tangles. So within the neurons and with dendrites that go out from the neurons, they have these neurofibrillary tangles. And we know now, Alzheimer didn't know it at the time, that these plaques are caused by a sticky protein in the brain. It's a naturally occurring protein, but what it does is it starts to aggregate and form these molecules of amyloid sticking together to form these clumps in the brain, deposited in the brain. And then this begins many years before the next phenomenon occurs of the neurofibrillary tangles caused by another misfolded protein in the brain called tau protein, TAU. And this tau protein misfolds and it disrupts the normal function of these of tau protein. It disrupts the normal function of the synapses between the nerve cells and ultimately the function of the nerve cells, and eventually they die. So this, based on evidence, is a model of Alzheimer's disease pathology. And you can see along the bottom axis there, starting on the left, normal, going all the way through preclinical Alzheimer's disease, through mild cognitive impairment, and then dementia due to Alzheimer's disease. So you can see here that Alzheimer's disease can begin long before a person has any cognitive symptoms. And it begins with abnormalities in various biomarkers. These are representations of the disease, biochemical changes and imaging changes which represent the underlying pathology that's occurring in the brain. So on the vertical axis, you can see the further you rise up the vertical axis, increasing abnormality of the biomarker. And so you can see with the red curve, the first one, this represents amyloid accumulation in the brain, and this is forming these amyloid plaques. And that can be measured in the cerebrospinal fluid, and it can also be measured by examining positron emission tomography scans, and we'll talk more about those. And then the next change is synaptic dysfunction. So the synapses in the brain start to be disrupted, they're not working properly, and this can be detected on two different types of imaging, FDG PET, which represents glucose positron emission tomography, PET scans using glucose as the tracer, and then fMRI, so functional magnetic resonance imaging, which is a different type of imaging. And then the next change to occur is tau-mediated neuronal injury. We can measure that in the cerebrospinal fluid by doing a lumbar puncture and obtaining a small amount of cerebrospinal fluid and analyzing it. And we have a lab in British Columbia with Dr. Mary DeMarco who does this. And I learned just yesterday that now I understand there's a federal program to actually support this. There was this initial announcement. It hasn't actually been implemented yet, but this is certainly coming. So there's greater recognition of this at a federal level. And you can see that curve rising, the green curve rising up to the left. And as you get synaptic function, you're now moving into the territory called mild cognitive impairment. So now these people are starting to get some symptoms. So now we're into no longer preclinical Alzheimer's disease, we've got symptomatic Alzheimer's disease at the level of mild cognitive impairment. And then beyond that, you can see impairment in cognition. So cognition is starting to decline. And you can see that, in the blue area, you're getting a change in the structure. So as neurons die, you get shrinkage of brain tissue. And so you can get reduction in the volume of the hippocampus and the enlargement of the ventricles within the brain. The cognition follows that in the purple line, and then eventually in the mustard colored color, there is decline in clinical function. This represents a pie chart by different types of drug development that's been advanced for Alzheimer's disease. So the first impression you get is there's many drugs in drug development. And if you look around the outside, there are some which are looking at disease-modifying biological compounds in the green area at the top. To the right, you've got disease-modifying small molecules. In the blue at the bottom, you've got neuropsychiatric symptoms. And in the orange, you've got cognitive enhancers. And as you go from the outside in, you can see there are phases. And this refers to the phase development of clinical trials. So phase 1 is very early testing in small numbers of healthy volunteers as a rule, and just really showing that there is safety, that this is a safe compound to be trying. Phase 2 is a larger trial, and this is where there are sufficient numbers to actually be determining the dose, the appropriate dose, and seeing if there is a increasing response to an increasing dose. So really testing that, and also again looking at safety. In phase 3, again, an emphasis on safety, but now what you're wanting to prove that there is so-called effectiveness or efficacy. So that's the normal phases of development in people. And we're going to focus on the small pie right in the middle of phase 3 of the green area, and in particular, one of the compounds there called lecanemab, right in the center of that pie. That one there with a highlight. So we'll talk about lecanemab a little bit later. Amyloid PET scans that can detect amyloid in the brain at a level that is clinically meaningful were first described in 2004 by Klung in Pittsburgh, and it detects the particular part of amyloid in the brain, not the soluble amyloid, which is starting to aggregate, but it actually measures the plaque that's in the brain. So you've got a good measure of plaque in the brain. Now, a negative amyloid PET scan, which doesn't show plaque, says at that point that it's unlikely this person has Alzheimer's disease, because you need to have amyloid and then later tau to actually be able to have the pathology that underlies Alzheimer's disease. And then what the PET scan can do, can detect this amyloid accumulation for up to 20 years before tau protein causing neurofibrillary tangles and the onset of symptoms. You can have, therefore, amyloid plaque detected on a so-called positive amyloid scan in cognitively normal people. And we'll talk about a trial that is looking at this later on. Or it could be people who have mild cognitive impairment. And these people are obviously at increased risk of developing Alzheimer's disease. They can be predicted to eventually develop Alzheimer's disease. Now, the traces that are used, the original one was the Pittsburgh B compound, PIB. It's a C11 compound. It only had a half-life of 30 minutes. We've moved to other traces listed there, and they have half-lives of 2 to 3 hours. So when a person has a PET scan, this tracer may be made here in London at our cyclotron and then transferred to the PET scanner, or it could be made in Toronto and driven or flown to London. And then a person lies in the PET scanner, the tracer is injected into a vein, it travels up to the brain. If there's amyloid there, it'll bind onto the amyloid, and then the scan is performed to actually measure the amount of amyloid that's in the brain. And this is measured according to a scale called a Centiloid scale, which goes from 0, no amyloid, to 100, a lot of amyloid. We can also measure amyloid protein in the cerebrospinal fluid. Now, everyone has some amyloid protein in the cerebrospinal fluid, What happens in Alzheimer's disease is that people have a slow but progressive reduction of amyloid A beta 42 in the cerebrospinal fluid. So I should explain amyloid A beta 42. Amyloid naturally occurring in the body, the parent protein that this comes from is a protein which is 90 amino acids long, and A beta 42 is a toxic amyloid protein which is being cleaved from the 90 amino acid protein by some enzymes, and then this gradually decreases in the cerebrospinal fluid as it's deposited into the brain. And there's a ratio that we can measure in the cerebrospinal fluid looking at two of these amyloid molecules, A beta 42 and A beta 40, and this ratio is even a better, more accurate way of measuring this. And so a reduced A beta 40 or A beta 42 ratio in the cerebrospinal fluid of people with mild compound predicts the development of dementia with a high degree of accuracy. More recently, in the last 3 years, we've been able to measure amyloid in the blood, and this is using a particular technique. It's a single molecular array technique, or Samoa technique, and this is able to measure decreases in A beta 42 in the serum or in the blood. Now, the ability to measure this, the concentration is much lower, and you see a lower decrease in this because in the blood there is amyloid which comes from other sites other than cerebrospinal, other than in the central nervous system. So amyloid comes from other parts of the body. So detecting a decrease in the blood, the decrease we're looking for is a lower decrease of about 10 to 20%, as opposed to in people who have positive PET scans in the cerebrospinal fluid, where we can detect a decrease of 40 to 60%. Nevertheless, this is an important development. We also have tau protein biomarkers. Normal tau protein brings together and stabilizes microtubules inside the neurons in the brain. That's its normal function. But in the presence of amyloid misfolded proteins, these abnormal A beta 42 amyloid proteins that are sticky and accumulate in the brain, you get this triggering of this, what we call excessive phosphorylation. This is where there are additional phosphorylation bonds within the tau proteins, and these cause them to tangle and develop these neurofibrillary tangles within the nerve cells, and then eventually the death of those nerve cells, the neurodegeneration of them. Tau PET has been able to measure tau in the protein in the brain since about 2014, and again there are different traces listed there. It's very sensitive and it's more specific for the diagnosis of Alzheimer's disease. And the tracer begins in the part of the brain responsible for the short-term memory. So we start to see this tau protein accumulating in the hippocampus and the subiculum. These are parts that we think of as being responsible for short-term memory, and that's why people having damage in this part of the brain present often with short-term memory. However, there are other presentations of Alzheimer's disease, and some of you may be aware of this. The typical one is the one I've just described, the so-called amnestic symptoms or forgetfulness symptoms, short-term memory problems, and that's involving the temporal lobe, the parietal lobe with the tau deposition, and in the area of the hippocampus and the subiculum. But there's also some people that present with language problems due to a so-called logopenic aphasia. That's the medical term for that. And that has a different accumulation of tau in a different part of the temporal lobe, particularly the left temporal lobe. And then there's a visual spatial impairment presentation of Alzheimer's disease. And these are people with posterior cortical atrophy or posterior parietal tau. So the point I'm trying to make is when the tau proteins accumulate in certain parts of the brain, because of that part of the brain has a different function, the way in which the person presents may be different. The most common way we see it is people presenting with short-term memory problems. So tau has also been investigated as a possible indicator to determine the progression of the disease, so a so-called prognostic indicator, because many people in the clinic say to us, well, what is this going to look like in a year, 2 years, 3 years, 4 years? This is the sort of indicator that might be able to help us predict that. This shows a picture of 4 different people. The first 3 from left to right have normal cognition. So the CN at the bottom shows that they're cognitively normal. And the one on the far right has Alzheimer's dementia. And you can see that in the top 4 frames, increasing amounts of tracer within the brain in a PIB amyloid scan. So this is a PET scan using PIB as the tracer, and you can see increasing amounts of amyloid protein in the brain. And the 4 frames in the lower portion, these are tau PET images, and these people here have, these 4 people have increasing amounts of tau to the point where the person at the right actually has sufficient tau in the brain for them actually to now have the clinical syndrome of dementia. So now I want to talk about the exciting news that we have from lecanemab. So this was released in the New England Journal in November 2022. And lecanemab is a monoclonal antibody. So when you see a compound having the designation MAB, that stands for monoclonal antibody. And this particular monoclonal antibody sticks to or has a high affinity to soluble amyloid protofibrils, which are thought to be the more toxic ones to the neurons than the single amyloid molecules, monomers, or the insoluble fibrils. So it sticks to those and it removes those. And for this particular trial, people between the ages of 50 to 90 who had underlying Alzheimer's disease and who were presenting either with the symptoms of mild cognitive impairment or mild dementia could participate in the trial. And to demonstrate whether or not they had the underlying Alzheimer's disease, they had to have evidence of amyloid accumulation on the PET scan, or alternatively, they could have a lumbar puncture showing that they had that biomarker A beta 42 to A beta 40 ratio that confirmed that their underlying cause of their cognitive impairment was Alzheimer's disease. And then they were randomized either to be 1 to 1, 50% in the treatment arm and 50% in the placebo arm. And they were then given intravenous treatments, which take about an hour to give them, at a dose of 10 milligrams per kilogram every 2 weeks. And this was an 18-month trial. And then if you do a clinical trial, you actually have to have endpoints. You have to be able to say, is there any difference between the people in the treatment arm and the people in the placebo arm to show that the compound you're testing actually makes a difference? So for clinical trials in Alzheimer's disease, the instrument that we use is something that's been developed called the sum of the boxes, or CDR-SB. And this is a instrument that's made up of input from the participant and also from the caregiver by asking them a number of different questions and getting them to look at different domains. And I can talk about that more later if anyone wants to know about that particular, but it is considered a really robust primary outcome measure. And this was actually determined through an observational study called the Alzheimer's Disease Neuroimaging Initiative, which we, as one of the 60 sites in North America, have been participating in since 2006. That study actually demonstrated that the CDR, sum of the boxes, was the most robust measure of capturing the natural course of Alzheimer's disease. So that Alzheimer's Disease Neuroimaging Initiative was a very important study, ADNI, and it continues, and I'll talk a bit more about that later. A secondary endpoints was looking at the change in the amyloid burden. So they were going to have people have a PET scan at baseline, and then you wanted to see, are we actually removing amyloid from the brain? And that was measured again at 18 months. And then they had some measures of memory, detailed test called the Alzheimer's Disease Assessment Scale, or ADAS-Cog14. There was a change in the Alzheimer's Disease Composite Score, ADCOMS, which is focused more on people with mild cognitive impairment than Alzheimer's disease, or rather than people who have dementia. And then there's the change on the Alzheimer's Disease Cooperative Study Activities of Daily Living Scale. So these are all of the secondary outcome measures there, those 4 secondary outcome measures, to prove whether or not this made a difference. So this is the first image, and you can see here that lecanemab significantly slowed disease progression on the clinical dementia rating scale by 27%. So let's try and interpret this graph. Across the bottom, bottom axis going from 0 to 18 is the months and the visits during those particular, at each of those points where measures are made, where the sum of the boxes clinical dementia rating scale is completed. And on the vertical axis, starting at the top, you've got 0 going down to 1.6. This is measuring the change in the sum of the boxes score from the baseline. So they look at your baseline sum of the boxes score and then looks at the change in that over time. Across the bottom, you can also see the numbers of people participating in the study. So at the left, in the black, you've got 875 people who are participating, and they are in the placebo arm, and then there's 859 in the lecanemab treatment arm. And by the end of the study, at the right, on the bottom there, you've got 757 who are in the placebo arm and 714 in the lecanemab arm. And then if you look above at the 2 lines, you can see that there are 2 lines going down, the green line representing people who received the lecanemab and people who received, who were in the placebo arm of the study. And you can see that those 2 lines are moving apart. They're so-called diverging. And you can see that they're diverging as early as 6 months. If you look at the 6 month there and that circle there, you can see there that they're diverging, and you can see that the error bars or confidence intervals bars don't overlap. So this means at that point, that is statistically significant. So that's what those 2 little dots are indicating. And then you can see the lines continue to diverge until you get out to 18 months. So this means that both groups are declining, but the group on lecanemab are declining less quickly than the people in the placebo arm. So this is a really important finding. This shows the ADAS-Cog. So this is also showing some divergence over a period of time using the memory scores, and over 18 months it's showing that there is slowing of disease progression using this memory score, the ADAS-Cog14 by 26%. And this now looking is at the activities of daily living scale. So again, same parameters, 18 months, and you can see that again, you got the divergence at 6 months. You can see that the 2 groups are separated there at 6 months, and then by 18 months, even more separated. And this is a difference of about 37% between the 2 groups. Now this is looking at the response to the amyloid. So again, across the bottom, 0 to 18 months. Now you can see that in the placebo and lecanemab groups, there are smaller numbers, 344 and 354. So not at all of the sites that were conducting the study was PET imaging available. It's not available everywhere. So you can see that, and then over the course of the trial, there are a smaller number, because there always, with any trial, there's always a certain number of people who drop out from the trial. Now you can see across the top, the black line, these are the people in the placebo arm, and so that line has a tendency to rise over time. So what you're seeing is that these people, instead of having less amyloid, they're having increasing amyloid, which is what you might expect in a placebo arm. Over 18 months, they would continue to accumulate some amyloid in the brain. But what is quite dramatic is the amount of amyloid lowering that you're seeing in the treatment arms. So this is quite significant, where there is a drop of measured in the centiloid scale. The difference between the two groups on average across the two groups is almost 60 centiloids. So they're going from about 80 centiloids at the top down to 20 centiloids in the treatment arm at the end of 18 months. And this is just to give you an image to see what this actually looks like. So on the left, we have the same diagram that we had in the previous slide, but in the right, you can see at the top, you can get the top three images, 3 cuts in one person at the baseline, and below those 3 images, the same person, 3 cuts through their brain using a PET scan, and you can see that this is 18 months. And you can certainly get the impression that there's more amyloid in the lower 3 than the top 3. And then if you go to the bottom 6, the top of the baseline, and you can see that the centiloid score at baseline is about the same in both groups. It's about 79 at the top, in the top group for placebo, and about 80 for the baseline for the treatment with lecanemab. And then if you go to the bottom 3, you can see there's much less amyloid in the brain, and this is measured at 22 centiloids. So the bottom, there are the summary there of the results, which just recaps everything that I've already said. But the conclusion from this is that lecanemab reduces markers of amyloid in early Alzheimer's disease, whether it's mild cognitive impairment or people with mild dementia, and resulted in less decline than placebo on all measures of cognition and function at 18 months. So all of the primary and secondary outcome measures were positive in favor of lecanemab, which was very compelling data. And these differences were observed as early as 6 months. And these findings encompass a broad range of endpoints and subgroups. So they've got different endpoints as I had described them earlier. Now, the next thing that I want to talk about here is some of the, we want to talk a bit about side effects. And there's microhemorrhages that occur in brains, and these are tiny bleeds that occur in brains. And the thing that is important to understand is that we all are at risk of developing spontaneous microhemorrhages. And another term for these microhemorrhages is amyloid-related imaging abnormality, or hemorrhage. And those are the sort of hemorrhages that you see as part of clinical trials. But microhemorrhages occur in the general population at about 5%. Now, people with mild cognitive impairment due to Alzheimer's disease are at increased risk, about 15%, will develop microhemorrhages. People with Alzheimer's disease more. People who have had what we call ischemic stroke, so people who have had a stroke where there's been a blood clot block a vessel, they'll have microhemorrhages in the brain as a consequence of that. And then there are people who have hemorrhagic stroke, where there is a bleed, a major bleed into the brain. And in these people, they'll have many microhemorrhages. And in these people, probably some of those microhemorrhages have become macrohemorrhages, causing the hemorrhagic stroke. So the point here is to realize that microhemorrhages do and can occur spontaneously in different populations with different prevalences. So this is looking now at the overall adverse event, because you want to know, if you're doing a clinical trial, what are the side effects? And people always ask us with a drug, what are the side effects? So we've got 2 columns here. On the left is the placebo group, and on the right are the people in the lecanemab. And again, you can see the numbers that were in the trial. And below that, you can see an N, which is the number of people, and a percentage. And across at the top, the very first line is deaths. So in the placebo group, there were 7 people that died who were on placebo during the course of 18 months. They weren't getting any treatment, they were just on placebo, and there were 6 in the lecanemab group. So, and percentage-wise, basically about the same percentage. Then they looked at what we call serious adverse events, or SAEs. This is the terminology that's used in clinical trials, and this is serious adverse events with area E. This is the amyloid-related imaging abnormalities or edema that we can detect on MRI scans, and these often is asymptomatic. The people have got no symptoms. There's another serious adverse event with amyloid-related imaging abnormalities due to hemorrhage. And you can see that in the lecanemab, the percentage is slightly higher with both of those with the serious adverse events. So you've got 0.8% and 0.6% compared to in the placebo group 0 and 0.1%. But there were some with even the placebo group who did have the hemorrhages, or one person anyway, with a serious adverse event. And then you can go down further to what's called treatment emergent adverse events. So this is people who have developed adverse events, and you can see treatment emergent events without any area or infusion related reactions. And so the percentages are about similar. So about 80% of people had some sort of, during the course of the trial, adverse event in the placebo group. So this is the importance of doing placebo-controlled trials, because even within the 18 months, 80% of the people in the placebo group had some sort of adverse treatment emergent event. And we'll go into what those events were. And then treatment leading events leading to drug withdrawal, some in the placebo arm, but more 2.9% versus 6.9% of people on lecanemab. And then treatment leading to drug withdrawal, excluding adverse events of a serious nature or of particular interest. Those were similar. So let's go to the next slide. And now this looks, you got it there? So these now are examining adverse events of special interest. So we've got the 2 columns again, placebo and lecanemab with the N numbers, the people participating in the trial as before. And we've got infusion-related reactions. So these are people getting a reaction to infusion. So you've got people in the placebo arm, 7.4% had some sort of local reaction at the skin, and 26.4% of people with lecanemab. Area E, this is detected on MRI scans. I'll show you some images of that later. And this is 1.7% in the placebo arm and 12.6% in lecanemab. So this shows that edema is occurring as a side effect of removing amyloid from the brain. And then area H, you're getting some hemorrhage in the placebo group. We indicated that people can get hemorrhages, but it's 9% of the placebo group and 17.3% in the lecanemab group. Other adverse events, you've got lower down, headache, a bit higher in the lecanemab group, falls, a little bit higher in the lecanemab group. And the rest of the things more or less bounce around between both groups, are about similar in both groups. So we'll go to the next slide. So this now shows from a different study how we understand what's happening in the brain when we try and remove amyloid from the brain. So this is using PET imaging, measuring the amyloid of the brain, and looking at this area E and area H. And this was done in a trial called the babenusamide trial. And this was done in people who were ApoE4 carriers. So these are people at high risk of developing these symptoms. So this is particularly looking at people
who are at greater risk of developing these symptoms. The ApoE4 gene, as most people may have, some have heard of that before. This is a gene that is distributed in the population. About 15% of the population have one copy of the ApoE gene, and 3% of Canadian population have 2 copies of ApoE gene, and this is a gene that increases the risk of developing Alzheimer's disease. So we've got 6 images here. On the top left, we've got a screening baseline FLAIR MRI scan. So that's what a FLAIR imaging MRI scan would look like in a person participating in the trial. In the middle image at the top, you've got a screening PIB scan. So this is what a positive PET scan would look like with some amyloid, and this is an axial view cut from front to back through the brain. And you can see the ventricular system in the middle, that dark area in the middle of the image. And then in the week 6, you can see at the top right-hand column, you can see some areas of little sort of fuzzy haziness, and you can see that there's a little bit of swelling there, and that the wriggly parts of the brain are not as apparent when you compare it to the screening baseline FLAIR image. And then by week 16, and this is using this FLAIR image again, you can, on an MRI scan, you can see that there are some very tiny microhemorrhages. You can point them at the front, particularly at the front of the brain. And you can see that 19 weeks, that the amyloid is much less on the PET scan. And when you go across, in the first image on the left, you can see that the edema has gone away, but on the one on the right at the bottom there, with the yellow box around it and the cursor on it, you can see these microhemorrhages there. So let's go on to the next slide. And what we interpret this is showing that at the baseline, PIB was elevated, as you can see at the top there, consistent with the burden, the amyloid fibrils, the plaques in the brain. And at week 19, and the one directly below it, you can see that there's been a reduction of PIB. And both of the reduction of the PIB is in time and in space is associated with the development of area E and area H. So area E, the hemorrhages, and the microhemorrhages, and the area E, which is the edema, and area H, with the microhemorrhages. These have occurred in response to the babenocimab treatment and the removal of amyloid from the brain. So it's a side effect of the treatment. So this suggests that area E and area H may be related to the therapeutic clearance of amyloid. These are some of the tau biomarkers, and now that we can measure tau in the brain as part of the lecanemab study, you can see here that in the image on the left, you can see a line that's trending upwards, and this is showing that in the placebo arm, you're having increasing amounts of phospho-tau in the cerebrospinal fluid. And you're seeing in the bottom green line, you can see that in the lecanemab trial that the tau protein in the cerebrospinal fluid is declining. So this is normalizing the cerebrospinal fluid. And in the one to the right, you can see the same pattern being mirrored in the blood. So the toxic tau, p-tau181 is increasing in the placebo arm, and in the lower green arm, the treatment arm, it's declining. So this is giving biomarker evidence, both in the cerebrospinal fluid and in the plasma, in the blood, that the lecanemab is doing what it's supposed to be doing by normalizing tau protein, both in the cerebrospinal fluid and blood. The biomarkers have the potential within clinical trials to actually be helpful in being able to help identify people
who might be appropriate to be considered in a clinical trial. So if you look at the left, you've got the general population, and you can have a biomarker, such as a blood biomarker, and this may be able to determine, is this person at risk or not? So you may have a group who are healthy in the black, and those who might have the biomarker for Alzheimer's disease. And then these are the people that can then go on further into a clinical trial, who may have other biomarkers, imaging biomarkers, which are much more expensive. It costs about $5,000 to do a PET scan, which are not clinically available. They are available through clinical trial budgets. And then you allow, the biomarkers can look at targets of engagement. You can see if the biomarkers are improving or getting worse, as I've shown with the tau biomarkers and the amyloid biomarkers in the lecanemab trial. And you can then tailor this to treatment outcomes to endpoints. You know, how long do you have to continue to give people intravenous antibody? What is the optimal duration of treatment? At what point can you stop and then just follow their biomarkers along and see whether or not they cognition is remaining stable or whether they're continuing to decline? And these future potential use for biomarkers would be in primary care. So a family physician, again, on the left, could have a population of people, some of whom may have a low probability of developing Alzheimer's disease, and obviously no need to worry, but others who may have a medium risk may need closer follow-up, and others who may be very high risk. And these biomarkers, once developed and validated in a diverse population, in a primary care population, then I think we'll see these biomarkers, as I've described in the clinical trials, actually in the primary care clinic, and people being able to get it in the community. The last bit I want to talk about is just some of the current trials. And one thing that we have been doing with Alzheimer's disease treatment trials is going from the original trials in mild to moderate dementia, moving back into mild dementia, back into mild common impairment, and now we're moving right back into people who have normal cognition, but who are at risk of developing Alzheimer's disease. And this is a treatment trial with lecanemab. So this is the A345 AHEAD study. There are 4 trial sites in Canada, we're one of them, and there are 75 globally. So this is a big initiative. And the A3 group are people who have very early preclinical Alzheimer's disease. So they are regarded as having an intermediate amyloid level in their brain with approximately 20 to 40 centiloids on a PET scan. And then the A4 study are for people who have preclinical Alzheimer's disease, and they have more amyloid in their brain. They have more than 40 centiloids in the scale. And that's going to make a difference as to the particular treatment regimens they have. One has got more amyloid, so maybe they would have a higher amount of treatment or longer treatment. And so everyone participating in the study are people who have evidence of amyloid on PET or CSF. They again get a 1-to-1 ratio intravenous lecanemab. They have to have normal cognition, be cognitively unimpaired, be ages 55 to 80, and those who are age 55 to 64 must have one other risk factor. So at least one copy of that risk gene, ApoE4, they could have 2 copies of ApoE4, and also known to be PET positive, either on a previous PET scan or maybe from another clinical trial, or cerebrospinal fluid that is positive. Or they may have a first-degree relative who's been diagnosed with dementia before age onset of age 75. So people who have a parent who had Alzheimer's disease diagnosed before age 75 and between the ages of 50 and 80, this may be of interest to them. It's a 4-year study. And the people who on the A3 arm who've got less amyloid in their brain, they have monthly infusions, and the people on the A45 study are people who have infusions every 2 weeks until 2 years, and then they go to monthly infusions. And they're going to be using primary endpoints. Now, think about this trial. These people all have normal cognition, so we're not going to be able to measure their memory scores. So there's the primary outcome measure is going to be looking at amyloid clearance of amyloid from the brain, but they're also going to have a new scale, a preclinical Alzheimer's disease cognitive composite. So this is a very sensitive measure to measure any changes that might occur from baseline over time. Another trial which has been conducted across Canada at a number of sites, but at Parkwood by our colleague Dr. Manuel Montario Dasso is doing the Synergic trial. So this is another trial using multi-dimensions in the way that the Finnish trial was done. And this study is looking at people who are age 60 to age 85 who have a diagnosis of mild cognitive impairment, and they have to have the typical criteria to make a diagnosis of mild cognitive impairment. They must have subjective memory complaints, objective evidence of impairment, preserved instrumental activities of daily living, and absence of dementia. And then they also have to have 2 or more dementia vascular risk factors, 2 or more risk factors, either low physical activity, a poor diet, insomnia, or one of the several vascular metabolic risk factors, so diabetes, high blood pressure, hyperlipidemia, smoking, those things. And then you can see there's a flow chart here where people will go through a screening assessment. They're going to try and randomize 550 people. So this is quite an ambitious study for this type of study, and they will be divided between the different sites. And there are 2 arms here. So on the left is the treatment line, where people will get online personalized intervention. So this is going to be done largely at home. So people won't have to travel into a site. They will have coaching sessions every 2 weeks, targeting their particular physical activity level, their cognition, diet, sleep, and looking at vascular risk factor management. In contrast, the comparison group will receive the online Brain Health program. This is an online program with education modules on physical activity. This has been developed in Canada, and it's got 181 modules, and it's got a lot of information there about cognitive stimulation, diet, sleep, vascular risk factor control. And then there's going to be a 6-month assessment and then a 12-month assessment. So this is a 12-month study. So this is half the length of the Finnish study. So what they're trying to show with this very intense coaching, is there a difference between providing people in the comparison arm with educational information versus an intense coaching sessions focusing on specific personalized risk factor management. We also have some new observational studies. The Alzheimer's Disease Neuroimaging Initiative is now going into another 5-year term, ADNI 4. And this is really going to focus on this time on recruiting underrepresented populations. One of the things we've found in ADNI is that there's an overrepresentation of white English-speaking and college, university educated people. And we want to focus on getting people that are underrepresented, and particularly from BIPOC and Spanish-speaking communities in the States. And we are going to engage in this as well. So this is something that we want to be able to contribute to make sure that these studies are much more representative of the population we have in Canada and in North America. The other study we're doing is the comprehensive assessment of neurodegeneration and dementia. We've been running this study now for 10 years. Our group at Parkwood Institute, the Cognitive Clinical Trials Group, has been the lead study for this national study, 30 different sites across the country. Our group have been responsible for training them. Dr. Jennifer Fogarty did all of the neuropsych training. Dr. Sarah Best did all of the management at the trial sites and coordinating with our other clinical trials coordinators and research assistants at our own site to be a very successful and lead site, recruiting a lot of people to participate in this observational study. And our focus this time round is to focus on people to try and address this issue of people who have up to grade 12 education with life experience, because this group of people are underrepresented in our current cohort in the 1,100 people that we already have participating in the Compass ND study. So I want to thank you, the people that have participated in our trial and all of our studies, whether they're observational or individual studies, and their study partners, because when you saw that diagram earlier on with all the clinical trials being done, there are a huge number of people that actually have to be able to participate in these studies to actually be able to bring this research to you, to be able to present these exciting results to you. So it's the only way we're going to be able to advance this. So I want to thank the people that we have had as participants, their study partners, and we welcome others to consider the opportunity to actually participate in these studies. This is just to acknowledge, the next slide just acknowledges all of the team that involves, as myself, Jenny Wells, and Dr. Bangu, one of our newest geriatricians in London, who actually will be leading the A345 study. They're already recruiting into that study. We've already got a number of participants starting to start in that study. And this is us during the pandemic. We don't have to wear the masks anymore unless any of us have got colds. Sarah Best is our research manager, who has been our leader of our group, along with Christy Coleman and all of our coordinators. Those who've been participating in trials will recognize the names of some of these coordinators who have been with us for a very long period of time and are able to give us the longitudinal follow-up and engagement with people. And then the other researchers, nurses, Dr. Fogarty I mentioned, and our collaborators across the city. So we'll stop there. We can go back. Yeah, okay. Thank you very much, Tara, for advancing the slides and keeping us on track.
You're welcome. Hopefully my pointing was okay with the the arrow there.
Very good, thank you. Yes, you're following that. At least I knew one person was understanding what I was saying.
I was focusing because it's a lot of information, but it's so fascinating to hear about all of the different studies that are going on and how promising, you know, some of them sound, right? So we're going to have a question and answer period. So if you want to type your questions into the Q&A, if you move your mouse or touch your screen, there's a Q&A button at the, on mine it's at the bottom of the screen. You can type in there, and Catherine is going to share the questions with Dr. Borrie.
There was one question that came through earlier on in the presentation. This attendee wanted to know what the difference was between lecanemab and donanemab.
That's a good question. These molecules developed by different companies. Off the top of my head, I can't give you the specific differences. They are both monoclonal antibodies, as all of these are. So they are monoclonal antibodies. The monoclonal antibodies target different parts of the amyloid, the different types of aggregations of amyloid molecules. I think donanemab and lecanemab are actually fairly similar in the molecules they target. That's in contrast to a molecule which was tested early on called solanezumab, and solanezumab targeted primarily the soluble monomers and dimers, the single amyloid protein, and it was able to show that they could remove that from the blood, but it didn't make any difference to removing amyloid from the brain, and it didn't make any difference to slowing the progression of the disease. Whereas I think we're going to be seeing information coming out from donanemab trial fairly soon. Certainly the phase 2 data was very promising, and we're hoping that donanemab is going to be positive. Gantenerumab was another trial that was published at the same time or presented at the same time as lecanemab, and it targeted a slightly different species of amyloid molecule and didn't have the positive effect in slowing the progression of disease. So I think it does, we're learning that it does matter as to which type of amyloid protein, which type of aggregation is targeted by these molecules.
Thank you. Next question. Clinically, do the patients who develop eczema and hemorrhagic infarcts show any symptoms?
But that's a good question. So what the question, if I can rephrase it, is that people who have the edema, in the brain, so the swelling in the brain swelling, and people who develop the little hemorrhages, the microhemorrhages, are they different? Do they have side effects? About 85% of the people who have these edema and microhemorrhages, this is detected on safety MRI scans, which are done after the first infusion, the third infusion. During the course of the trial, these safety MRI scans are detected. And so about 85% of the people in whom we detect these changes, they have no symptoms at all. They're asymptomatic. They don't know that this has actually happened. There were some that you saw who had headache. Headache was more common in the lecanemab, and those would represent some of the people, the 15% who actually did have symptoms from the infusion of lecanemab. And part of this might be a dose effect. I mean, one of the things we're going to learn is what is the dose, because what you could see from those images is that the amyloid is removed really early on in the treatment. You know, within 1 or 2 doses, you're starting to remove amyloid quite aggressively. And so this is going to be part of the potential regimens that eventually I anticipate come. The rate at which a person is given amyloid may depend on how much amyloid is in the brain and how quickly it's removed from the brain. So I think these are refinements that are going to be developed over time. But both aducanumab, which was another earlier, somewhat controversial intravenous antibody, particularly because of the way it was, the approval process went, and lecanemab have now been approved for clinical use in the States. Not yet in Canada, and we have to wait and see what's going to happen with the FDA's decision next month on lecanemab. Are they going to go to full approval, because it's got conditional approval now? And then that likely will lead Eisai, who's the company makes lecanemab, take it to Health Canada seeking approval.
Thank you. Next question. Have you heard anything about a drug for dementia patients called methylene blue?
I have, and there was a lot of excitement about this. The first time I heard about this was about 15 years ago at one of the Alzheimer Disease Association International Congress. And there was a lot of interest in it at that time. One of the issues about giving people methylene blue as a treatment is that it makes your urine go blue. So it meant that if you had people in a placebo arm versus a treatment arm, it became fairly apparent who was on the treatment and who was not. And the idea of blinding is that you try to reduce the likelihood or eliminate the likelihood that people know. So for that trial, they had to give people in the methylene blue placebo arm a little bit of methylene blue so that their urine turned blue as well, so that you couldn't tell the difference between the two groups. However, I haven't seen further advances with that in terms of it, and it's probably on that pie graph. I haven't worked out which one it is, but it is. But I have heard of it. There was a lot of excitement, but I haven't seen further advance of that in terms of a phase 3 trial that through C5R. C5R is the Consortium of Canadian Centres of Cognitive Clinical Research. So any trial that is going to be brought to Canada. So not all trials come to Canada. We've got 4 sites for the lecanemab trial out of 75 globally, and there's many countries involved, maybe 20 or more countries involved with that trial. It's possible a methylene blue trial, and I think it was a Korean group, if I'm not mistaken, were developing it originally, maybe developing it or trialing it in a different area. But I haven't seen it at this point as a phase 3 trial, and I haven't seen any data on it yet, but I have certainly heard of it.
Thank you. This next question, hypothetical situation, individual 40 years old, noticing potential cognitive changes. 24 on a MOCA test a couple of years ago. It looks like there's some Alzheimer's disease in this person's family. Just this attendee is looking for maybe next steps.
Thank you for that question. So can I just clarify, was the age of the hypothetical person 40?
Yes.
Okay, so 40 is a young age for Alzheimer's disease. However, it's not unheard of. So let's explore that a bit. There is a genetic form of Alzheimer's disease which is rare, and it does occur in some families, and it's a mutation in commonly one of 3 genes, presenilin 1, presenilin 2, or the APP gene. So they get the genetic mutation in those genes. You get 2 copies from you each have 2 copies of every gene in your body, 1 from each of your parents. And in some families, that gene, that mutation in that gene, guarantees that a person will get Alzheimer's disease, and it will depend on the age at which that person developed cognitive impairment. So if the parent, so when we're talking about family history, as in this case, if the parent had developed cognitive impairment at age 40, and this is an offspring of that relative, then you would have to wonder, could this be a person who has a presenilin 1 mutation, APP, or presenilin 2 mutation that might be causing the Alzheimer's disease? Now, I must emphasize that this is rare. I think in my career here at Parkwood, I think I've seen 4 families here. So this is most uncommon.
And the ages, sorry, the ages of onset in the family, in the older family members, were 74 and mid-60s.
Yeah, so this would be unusual that this person would have Alzheimer's disease at 40 if the family history was 70s and 60s. That you might expect them to have Alzheimer's disease at a later age, maybe in their late 50s or early 60s, have symptoms at that time. So you'd have to ask, are there other factors? So could this not be Alzheimer's disease? Could it be some other thing? Could depression be a factor? Are there reversible causes of cognitive impairment in this person? So a family physician would start with blood work and look at thyroid function, and look at vitamin B12 level and calcium levels to see if there's anything else. Has this person had head injuries through their life? So there is chronic traumatic encephalopathy, which occurs in people who have had repeated concussions in their life. So this puts them at an increased risk of developing Alzheimer's disease. Are there other things, like are they on medications? Are they drinking alcohol regularly? Could this be a factor? Are they using other drugs? That could be a factor. So looking at all of these potentially reversible things, the person should have some sort of neuroimaging, rule out things like a brain tumor. If they've got no other symptoms, headache or any focal neurologic signs, less likely, but nevertheless, one would want to do that type of imaging. And then I think if there was evidence of progression, particularly you repeat the MOCA, because a single MOCA does not make a diagnosis of Alzheimer's disease. If you do the MOCA on that person again, and now their MOCA is 28, now their score is in the normal range. And maybe there were other things that were going on in their life, stresses, losses, that might have been contributing to cognitive impairment. So I think I would certainly look for those things there, but I think it's an excellent question to explore some of those issues.
Thank you, Dr. Borrie. This next question, the attendee writes, both of our children are in the 45 to 50 age group. Is it reasonable to recommend testing based on one of the parents being diagnosed with dementia?
So one of the parents diagnosed with dementia, what we didn't hear was the age of the person of the dementia. So if they're in their 40s and 50s, one might make an assumption that the parent could be somewhere between, they have to be at least...
they just clarified that the parent's age is 72.
72, okay, thank you for that clarification. I think the question is, what sort of testing would one do in that instance? If they've got absolutely no symptoms at all, one wouldn't need to do any testing. They're too young for any clinical trials that would detect amyloid. If they were 55 and over, then certainly they could participate in the A4-5 study, because with that particular study, you get screening for Alzheimer's disease. So you get a blood test first. If the blood test is negative, you don't go on to get a PET scan, because a PET scan is $5,000, and then if it's unlikely that you're going to have an amyloid PET scan, they're not going to go on and do a PET scan. They're going to look at the blood biomarker, because blood biomarkers are getting very good at predicting who's going to be positive on a PET scan. But if they were 55 and over, then that would be a way to get a free blood screen, a very simple test. If it was positive, getting a free PET scan, and then deciding, depending on the result of that, if it was positive, do I want to do something about it? Do I want to go on, do a clinical trial, or do I want to wait and see whether I'm going to develop symptoms? So there's choices. I mean, any clinical trial is optional. But I think at those ages, 40 and 50, unless they were having some symptoms. Now, all of the other information that would be important, what I would encourage them to do, though, given that the family member had symptoms, their diagnosis in the early 70s, but maybe symptoms sometime in the 60s, all of those non-drug approaches that we talked about and the control of the vascular factors, the 20 different things that we did, there's lots of things there that can be done that anyone should be doing who may be at risk. Anyone who wants to preserve brain function should be doing all of those things, and reducing the likelihood of progression to dementia. Because if we can reduce the likelihood of progression by paying attention to those risk factors, if we can reduce that progression from mild impairment to dementia by 5 years, we reduce the prevalence of dementia around 66%. And if we can delay the progression from mild impairment to dementia by 10 years, the likelihood of dementia, the prevalence of it would be reduced to about 93%. And you ask why? Well, people are going to do the decent thing and die of something else. They're going to, because most people die of cancer, heart disease, stroke. They don't die of dementia. It's about the fourth most common cause of dementia. So one can still really enhance one's quality of life, do all of those things, and then we're all going to die of something, but we don't have to die of dementia.
Thank you. Next question. How is it someone has dementia or mild cognitive impairment as a diagnosis, but the brain CT scan is normal with no issues?
Okay, that's a good question. The CT scan may look normal, may be reported as normal. What it means is that at that point in time, you remember those curves that we showed earlier on, it's just not at the point where the atrophy is actually starting to occur, because the change on the brain as a volumetric change in the brain is a late sign. That occurs, so you can have people who have a normal looking brain, but they don't have the atrophy in the hippocampus, they don't have the enlargement of the ventricles, they don't have the shrinkage of the gyri of the brain, they don't have that shrunken walnut look. It looks normal. even though they have a progressive cognitive impairment. And it's just that at that point, they haven't had sufficient shrinkage for it to show up on an image. So the shrinkage is a late sign, and usually a person has to have significant dementia sometimes before you'll even see it. So it's certainly possible that a person with mild cognitive impairment has a completely normal image, because it's a later sign. It's not an early sign.
Okay.
Does that make sense?
Yeah. Thank you. Next question. Is London one of the 4 sites in Canada? I assume they mean the
yes. So the 4 sites, there is one in London, there are 2 in Toronto, and there's one in Kentville, Nova Scotia.
Thank you. And then just a comment, thank you for your helpful response to one of the questions. And then 2 last questions. Number 1, is lecanemab only indicated for mild dementia, or can it help someone in the moderate stage also? So in this situation, the MoCA score was 7 out of 36 months ago.
So a MoCA score of 7, as you would appreciate, is quite low, and this is certainly consistent, doesn't make a diagnosis of moderate dementia, but would be consistent with that level of dementia. The trial with lecanemab was done in people with mild cognitive impairment and mild dementia, and so it wasn't tested in people with moderate dementia. And I think you have to think about, with moderate dementia, a person with moderate dementia will have the atrophy in the brain, they will have the shrinkage in the brain, and that shrinkage is because those neurons have been lost. The neurons have gone away and they're not coming back. And that, I think, what we're finding, and from the trials that we've done over many years, that treating people with moderate dementia to try and slow or reverse the disease, it's too late. We really need to be starting much earlier, and what you've seen tonight is that we're now going even earlier than mild cognitive impairment. We're seeing people with normal cognition who are at risk, because what we're trying to do there is to preserve nerve cells before they get damaged, before the tau tangles take over and kill the nerve cells. But with moderate dementia, the focus is different, and is to try and preserve quality of life. We do that through our programs, through the Alzheimer societies, through the McCormick Home Day program. But I'm afraid it's beyond that point. Hopefully we can advance these treatments so that we can get people and preserve brain, and so that people don't have this progression through to moderate dementia and further.
Mm-hmm. And then one last question is L-citrulline. I'm not sure if I'm pronouncing that correctly. Is L-citrulline supposed to be helpful in dementia?
To answer that question, I would have to look at the literature to say, is there a randomized controlled trial that shows that this makes a difference? And I think that particular compound may be a supplement, or it may be a health product. I'm not aware of it being, certainly not an FDA approved or Health Canada approved compound to slow or reverse dementia. A lot of health products have these claims. They have claims on the bottles. They have very fancy looking bottles, fancy advertisements on television. And it's unfortunate, but these don't stand up to the scrutiny of a randomized controlled trial. So I hope people tonight have got some idea of why we emphasize the importance of a randomized controlled trial. And if the makers and people producing this particular compound want to demonstrate its effective, they need to do the gold standard and do a randomized controlled trial and show that it actually either helps the symptoms of the disease or hopefully slows the progression of the disease. But that is the standard we have to hold our treatments to.
Thank you. Well, last call for questions, folks.
Great questions. Yeah. Yeah, very interesting.
Thank you for your questions, and thank you for your interest.
Thank you, Dr. Borrie.
Oh, you're most welcome. Thank you very much, Tara. Thank you, Catherine. Thank you for Shelley at this end for helping us with with the technology.
Yeah.
Oh, one last question. Hang on. Oh, just very helpful and thank you. Lots of thank yous coming through.
Okay.
From our participants.
Wonderful.
You're most welcome.
Thank you, Dr. Borrie, and we will not wait as long this time to have you come back and present for us again.
Okay, you're most welcome. Thank you so much.
All
Good
right.
night now.
Take care,
Have a good night.
everyone. Good night.